Hi, this is Dr. Ippolito, and this is the Chapter 8 lecture on cellular respiration. We'll be discussing the way eukaryotic cells undergo the breakdown of sugar to produce energy. Previously, we talked about photosynthesis, the process by which plants, algae, and some bacteria utilize energy from the sun to create high-energy compounds, including sugar. In this chapter, we'll learn how heterotrophs, or organisms that eat their energy, take that sugar produced by photosynthetic organisms and use it for themselves. You will notice several themes from Chapter 7 present themselves again, so Chapter 8 also includes variations on these themes. Essentially, cell respiration is breaking stuff to build stuff. The word respiration itself is the process by which organisms exhale or produce carbon dioxide while bringing in and utilizing oxygen. So cellular respiration is essentially burning sugar and producing carbon dioxide. The overall goal of cellular respiration is to extract usable energy from organic molecules. For today, we're going to show how we use sugar to make ATP, but it's important to keep in mind that not just sugar can be used in cellular respiration, but any other high-energy organic molecule that you eat, including proteins, lipids, carbohydrates, and even nucleic acids. So, today we're going to start with glucose, the 6-carbon hexose, and we're going to end with chemical energy plus carbon dioxide and water as side products. Another way of saying all this is we're going to oxidize glucose to carbon dioxide and reduce oxygen to water. Here's the overall chemical equation for cellular respiration. You can see on the left we start with glucose, C6H12O6, and we add oxygen, 6 oxygens, and that's molecular oxygen in a gas. And we convert these to carbon dioxide, 6 molecules of that, 6 molecules of water, and some energy. So you can see there's two separate reactions. There's a redox reaction and an oxidation reaction. Okay, so when we say redox, remember there's always the reduction and the oxidation. So glucose going to carbon dioxide is the oxidation reaction, and remember oxidation is the loss of electrons. So glucose is losing its electrons. And where are they going? Well, ultimately they will go to water. So another way of looking at this is glucose loses electrons, and along with that loses energy, and it puts those electrons into oxygen. And oxygen is the final electron acceptor in this overall process. Deceptively simple. Cells slowly break the glucose down, releasing its chemical energy in the process. The more you carefully pull something apart, the more useful energy you can get out of it. Another way of saying this is, the slower and more carefully you take something apart, the more useful things you will get out of it. One funny analogy I sometimes talk about is watching Cookie Monster eat a cookie. When Cookie Monster eats a cookie, he eats it really fast and he's really messy. And of course he gets no energy out of it, because he's just a muppet. If you eat a chicken wing really fast, and you chop some of the meat off in the easy parts and throw it away quickly to grab another one, you can realize that there's lots of energy in that original chicken wing that you didn't get from it. Now imagine you're starving and you have one chicken wing. Think of how carefully you're going to eat that chicken wing. You're going to pull it apart and even possibly break the bone in half and suck some marrow out. As gross as that sounds, you can see that you would definitely extract more energy from that chicken wing if you're careful and slow and methodical than if you eat it quickly. So cells have evolved very complex ways of slowly burning sugar, many steps along the way, and extracting as much energy out of it as possible. And it turns out that with the care of glycolysis and oxidative respiration, about 40% of glucose's chemical energy is converted into ATP, assuming everything goes well. Where's the rest of that energy? Well, it gets given off as heat. That's why you have a high body temperature. If things don't go well, in other words, if you don't have oxygen, only 2.1% of the energy from a single sugar molecule is extracted into ATP. This is too inefficient to survive. And if you don't believe me, try holding your breath and see how far you get. The tools we're going to use along the way here include two dinucleotides, nicotinamide adenosine dinucleotide and flavin adenine dinucleotide. You've already seen NAD someplace else in the form of NADP. Okay, so the only difference between NAD and NADP from photosynthesis is that extra phosphate. And just remember P in plant, or P in photosynthesis. Okay? Now, it also makes sense, if you think about this from an energetics perspective, that plants exist at a higher energy level and can hold things with a lot of energy. So any other nucleotide along the way, such as what happens in cell respiration, it can't hold as much energy. And so you'll see the reduced form of NAD+ later is NADH. NADH exists at a lower energy level than NADPH. And if you remember phosphate, what we talked about a lot was phosphate was able to store a lot of energy. So plants use NADPH because it's capable of holding much more energy than simply NADH. Flavin adenosine dinucleotide, flavin adenine dinucleotide, also holds even less energy than NAD. So if you were to put these in a spectrum of greatest amount of energy to least amount of energy, you would see it like this. And it turns out, by the way, FAD holds 2 protons. Okay? So that's the energy spectrum on how much energy these dinucleotide tools can hold. And again, remember, the point of these is to carry energy within a cell in a useful way. So if this is a cell, and this is NAD+ with its energy, NADH, okay, it's basically, it allows energy to move in a cell without it diffusing into the environment. Okay? Think of a cup of water that you put in a microwave and it's steaming hot. You can walk from one area of the room to another area of the room with minimal heat being given off from it. It's still hot over here. Okay? Whereas if you just turn the microwave on by itself and you allow that energy to dissipate into the air, well, you can't do very useful things with that, can you? So, dinucleotides are useful in that they carry energy distances to places where the cell needs them. And another way of saying these are their coenzymes. Both of them accept high energy electrons from the breakdown products of glucose. These electrons are ultimately used to build a proton gradient, just like in photosynthesis. NAD+ takes 2 hydrogens away from its substrate. One of those hydrogens carries an extra electron, and the other has none. The H- and the NAD+ combine to form NADH. The H+ is then released into the surroundings. By the way, this is not something that you don't have to study this. This is just FYI. FAD accepts 2 hydrogens and the electrons it carries. FAD is built from vitamin B2, or riboflavin. That's why we need vitamin B2. Okay, again, this is not a slide you need to study. This is just FYI. Overall, there are 4 phases of cellular respiration. We're going to go through each one of these in detail. Glycolysis, the preparatory reaction or preparatory phase. Sometimes it's called the prep reaction. The citric acid cycle, also known as the Kreb cycle, and the electron transport chain. or oxidative phosphorylation. Fermentation occurs as an alternative to these guys. So in other words, cells can either go through glycolysis and then fermentation, or they can go through glycolysis and then all of this. And we'll talk about this in more detail. Here's a mini map of the mitochondria. As you can see, steps 2, 3, and 4 occur inside the mitochondria. Step number 1, glycolysis, occurs outside the mitochondria, in the cytoplasm. Again, we'll talk about these more in detail. So step 1, glycolysis. Glycolysis is when glucose is broken down into 2 smaller pieces. 2 ATP are invested into this process to, quote, get the ball rolling. Okay, imagine you have a ball at the top of a hill, and it's got a lot of potential energy because it's high off the ground. You kick it a little bit. That requires energy. However, the amount of energy the ball gives off on its way down is substantially more than the energy you put in. In the same way, we're going to invest 2 ATP in the process of glycolysis. We're going to yield more than 2 ATP. We're going to actually make 4 ATP. So you put 2 in and you get 4 out. So you net 2. That sounds like a good deal, right? If I said to you, I'm going to give you $2 and you're going to give me $4, I'm going to keep making $2 every time. That's a good deal. The energy from these pieces that we're going to talk about later are slowly extracted further. Okay, those pieces called pyruvate. I'm sorry, let me say that again. The energy from these 2 smaller pieces will be extracted and put into NADH and ATP. And of course a little bit of water is produced. At the end of all of that, after the end of glycolysis, glucose is broken down into 2 pieces called pyruvate. Pyruvate is a 3-carbon molecule. So we're going to make 4 ATP and we use 2. So we say we net 2 ATP as well as 2 NADH. Now I want you to keep track of these NADHs. They are the tickets to put in a machine that makes more ATP at the end of all of this. By the way, you can see at the top here, this yellow line indicates that glycolysis occurs outside of the mitochondria. The next phase is called the preparatory reaction, okay? And it only occurs in the presence of oxygen. So cells have to have a high concentration of oxygen present in order for the preparatory reaction to occur. The prep reaction is when pyruvate is pumped from the cytoplasm into the inner matrix of the mitochondria. Without oxygen, NADH reduces pyruvate to either lactic acid or ethanol and carbon dioxide, which we call fermentation. Again, we'll get to that later. Inside the mitochondria, a huge enzyme attaches a 3-carbon pyruvate to a coenzyme called coenzyme A, or CoA. In this process of attaching the 3-carbon pyruvate to CoA, carbon dioxide is released in the process, okay, thereby releasing the first carbon of the original 6. Okay, so we had a 6-carbon glucose molecule that got broken down into 2 3-carbon molecules. Okay, each one of these pyruvates is then attached to CoA, which is just a 2-carbon molecule left attached to CoA, plus a carbon is given off. This is the first carbon dioxide that goes into your into your respiration, that you breathe back into the environment, that goes and performs processes in photosynthesis. Okay, and of course this happens twice. Why twice? Because the 6-carbon glucose is broken down into 2 3-carbon molecules. Keep that in mind as you go through this. In addition to the carbon dioxide production, the energy that was released in this process is put into an NADH molecule. This is a picture of the pyruvate dehydrogenase complex. The Krebs cycle occurs also inside the mitochondria, and the initial substrate is the acetyl-CoA, which is the product of the prep reaction. The acetyl group is essentially the two carbons left over from the pyruvate attached to the CoA. During the Krebs cycle, the energy inside acetyl-CoA is slowly extracted. High energy electrons are given to NAD+ and FAD, along with some protons, and the now reduced NADH and FADH2 are used in the next phase, oxidative phosphorylation, which is an electron transport chain, similar to the one we saw in photosynthesis. For every turn of the Krebs cycle, 2 NADH molecules and 1 FAD molecule are produced per cycle. So the question is, how many cycles occur per glucose molecule? Okay, so one glucose molecule, which is 6 carbons. Again, remember, you have broken down to 3 carbon pyruvates, which then get broken into 2 carbon acetyl groups. Okay, so per glucose molecule, there's 2 Krebs cycle events happening. Okay, so in addition to the 2 NADs and FADH2s, actually 1 ATP is produced per cycle. So that was a typo. Okay, and 2 carbon dioxides are produced per cycle as waste. 2 carbon dioxides, that's the original 2 carbons from the acetyl group. Okay, so after the Krebs cycle, all 6 carbons from the original glucose are now completely converted back into the carbon dioxide they were built from. Okay. The fourth and final phase is called oxidative phosphorylation. This is the electron transport chain, and this is where the real payoff is. All of the NADH and FADH2 that has been generated up to this point give up their high energy electrons to an electron transport chain, which is embedded in the inner mitochondrial membrane, which extends out into the matrix space as cristae. Okay, so this is a cristae, and the electron transport chain is embedded inside the membrane. The energy from the NADH and FADH2 is used to pump protons into the mitochondrial lumen. In other words, the energy molecules out here pass their energy to the electron transport chain, which pumps protons in to this blue area. The blue area would be similar to the thylakoid membrane in photosynthesis. And the rule of thumb is, for every 1 NADH molecule that hands its energy off the chain, there's enough energy in the chemiosmotic force produced by the protons to create 3 ATP. For every FADH2 molecule that pumps energy in, there's enough left over to make 2 ATP. Okay? By the way, we're going through an overview. You will see these processes again. Okay, this is just an overview of the details of the mitochondria. So you can see the inner matrix is this gray area. The cristae are these areas here. In this example, you can see there's 1, 2, 3, 4, 5. Okay, circled in red are the cristae. You can see the inner membrane, okay, which is this area here. This is where the electron transport chain is embedded, and the ATP synthase we'll talk about later. You can see the lumen or intermembrane space, the blue, and you can see the outer membrane, which doesn't really come into play here. As I said earlier, protons are pumped into the shaded area, and ATP synthase is used to create ATP. Just like in photosynthesis, the protons will pump downhill, and in the process, ADP will be made into ATP. Here is a nice flowchart overview of the entire process. So you can start up here with glucose, and glucose gets funneled into glycolysis. Okay, one word about glycolysis: it is the most ancient cellular metabolic reaction known. Bacteria, all the way up through the animal, plant, and fungus and proteus kingdoms, all use glycolysis, with very few exceptions in the bacteria and archaeans. It's the most fundamental system that exists. All the enzymes in glycolysis are identical between everyone that uses them. In other words, your enzymes in glycolysis are the same ones used by bacteria. Another piece of evidence of evolution. So, the products of glycolysis are 2 pyruvates plus oxygen. I'm sorry, oxygen is not a product. Let me start that over again. Glycolysis takes glucose and breaks it down into 2 molecules of pyruvate. In the presence of oxygen, pyruvate is pumped into the mitochondria, where it undergoes the prep reaction. The prep reaction produces an NADH molecule, as well as acetyl-CoA. Acetyl is simply the 2 carbons left from the 3 carbons that came in from the pyruvate. Acetyl-CoA is fed into the Krebs cycle, which produces ATP, 1 ATP, 2 NADHs, and 1 FADH2 per crank per each acetyl-CoA molecule. CoA acts as the shuttle. It's a coenzyme. It's not used up in the reaction, but just keeps ferrying acetyl groups from the prep reaction to the Krebs cycle. The FADH2 and the NADH feed their high-energy electrons into the electron transport chain in the inner mitochondrial membrane, causing a series of redox reactions that pump protons. Those protons build up force to then generate energy for ATP synthase to create ATP. In the absence of oxygen, pyruvate is directly reduced by NADH produced in glycolysis. We'll get to the details of that later. Details.